Cancer Genome Atlas results, in which you can see that the patients who have hormone receptor positive cancers, which by gene expression profiling typically are classified as luminal A and B, and they appear to have distinct genetic features compared to those, for example, that are triple negative, which is primarily represented by the basal phenotype, the basal gene expression pattern. For example, you can see that PI3 kinase mutations are seen more commonly in luminal A and B compared to basal-like. And this gives us some glimpses that hormone receptor positive cancers may be different, and this information could in fact point to specific therapies. We also know, as you heard from Dr. Apro, that there are biological differences in hormonally sensitive cancers based on prior therapy. And this also has been confirmed by next-generation sequencing. This is work from Matthew Ellis and his colleagues, where they profiled several tumors that were known to either be aromatase inhibitor sensitive or resistant. And you can see by these plots, especially the inner part of the circle represents translocations. And you can see that the aromatase inhibitor resistant tumors, these are human tumors, were actually much more chromosomally aberrant, had many more chromosomal translocations, which sets up a greater opportunity for clinical resistance. If you look at the history of hormonal therapy that goes all the way back to over 100 years ago, when Sir George Beatson, a Scottish surgeon, noted clinical responses in women who had undergone an oophorectomy. And of course, this was well before the discovery of estrogen and estrogen receptors. Shortly thereafter, numerous hormonal therapies were adopted, including tamoxifen and aromatase inhibitors. But only recently have we seen the benefit of combining two drugs together to block hormonal pathways more efficiently, and that is with the approval of HER2-targeted therapies, specifically for patients whose tumors are both estrogen and HER2 positive, and then more recently from the approval of everolimus, the mTOR inhibitor. So let's first start with the hormonal therapies that we know are to be effective in advanced breast cancer, and ask the question, are there combinations of these hormonal therapies that can more effectively block the response and have a clinical impact? And the answer, of course, really came quite some time ago with the combination of ovarian blockade along with tamoxifen, where the combination was tested in several clinical trials. This is one of the larger trials to show that combination therapies actually resulted in improved outcome compared to tamoxifen alone. More recently, there have been attempts to combine fulvestrant, the estrogen receptor downregulator, with anastrozole, and here the results are not quite so clear. The first of these studies to report, the FACT trial, took patients who were both in first-line therapy. Most of these patients had previously received adjuvant therapy, so some of them may have met the qualification for being primarily resistant. And this study showed no difference in progression-free survival. On the other hand, dual hormonal therapy, the SWOG 0226 study, did show a difference that was statistically significant in survival, as well as progression-free survival from combining anastrozole and fulvestrant. The difference in this study is that most patients were previously untreated in the metastatic setting. In fact, many patients were de novo. They presented with metastatic breast cancer as their first presentation. So this population is different and provides a hint that maybe for unexposed patients that dual hormonal therapy could be more effective. I think these results are too preliminary and would need to be confirmed before we use these in routine practice, however. Another study, the SOPHIA trial, looked at dual hormonal therapy, this time in the second line. So this is all patients previously exposed, and found no difference in any of the outcomes. So if anything, this might only apply to initially treated patients. Our understanding of the pathways involved in hormonal therapy has changed quite a bit in the last few years. We now know that growth factor receptor pathways seem to modulate the effect of estrogen receptor. So signaling through receptor tyrosine kinases like HER2, EGFR, the insulin-like growth factor receptor, can actually phosphorylate the estrogen receptor and activate it. And this has led to significant interest in blocking these pathways as an adjunct to treating hormonal therapy. Also, directly affecting the cell cycle machinery has been tested. And then finally, looking at epigenetics, the methylation of DNA that controls the expression of certain genes. It has been shown that hormonal resistance can also be affected by changes in the chromatin and in the methylation of DNA. These are both events that affect the expression profile of cells. Let me first start with the pathways that we understand to be aberrant. In hormone receptor positive cancers, the most frequently altered gene from next generation sequencing is the PIK3CA gene. This is the catalytic subunit of PI3 kinase. Particularly the alpha subunit is the one that harbors most of the activating mutations. There are other mutations along the growth factor receptor pathway as well that could be targeted to benefit hormonal activity. These are known mutations in addition to the one I just mentioned, PI3 kinase. There are mutations in P10, AKT that are not as infrequent. And then, of course, there's amplification and mutations in the receptors themselves. And on the right side, you can see numerous pharmacologic agents that can target different aspects of these pathways. So all of these drugs are candidates to look at to see if we can augment the effects of hormonal therapy. Let me first start with the receptor blockade. This would, of course, would only apply to if HER2 is being blocked to the treatment of cases that are both HER2 and estrogen receptor positive. And I showed these slides earlier. The addition of trastuzumab to anastrozole, as well as the addition of lapatinib to letrozole, can improve outcome. However, you can see that the natural history of these patients is much worse than the HER2 negative. Here we see median times to progression that are very short, on the order of 2 to 4 months. But what about blockade of other aspects of the pathway in HER2 negative? So this was a study of patients with ER or PR positive, HER2 negative. This is very well known to you, a pivotal study that took patients in the second line after progression on aromatase inhibitor and randomized them to placebo or everolimus. And this study has shown a dramatic improvement in progression-free survival. The final 18-month follow-up was recently published, presented, and this significant improvement led to approval of this drug in many countries. However, at the final survival analysis, there is still no difference in survival. So a significant delay in progression-free, but no difference in survival. Moreover, it has been difficult to dissect apart which patients might respond. You might predict that if you have a mutation in PI3 kinase that is activated upstream of mTOR, that blocking mTOR should be particularly effective in that group of patients. However, we did not see that in analysis of the Bolero-2 study, where primary tissue was available for analysis, and one looks at the several mutations that can activate the PI3 kinase pathway. This would be activating mutations in PI3 kinase or loss of P10, both of which activate that pathway. It did not seem to matter in who benefited. So we still don't understand this pathway well enough to say, if you have this mutation, you respond to this drug, even though theoretically it should. Newer drugs against other parts of the pathway, directly inhibiting the PI3 kinase pathway, would also be predicted to be helpful. However, in one study that was presented at the San Antonio meeting just last month, there was no difference from the addition of pictillisib, which is a PI3 kinase alpha-specific inhibitor, when added to fulvestrant. This study showed no significant difference in disease-free survival. There was another study in the preoperative setting that also looked at this drug, a short 2-week exposure, to see if one could drop the Ki-67 score. As you heard from Dr. Apro, the drop in Ki67 has been found to be a predictor of longer-term outcome, and it is being used in window of opportunity studies such as this one to see if a brief exposure can drop Ki67. And what this did show is that there is a biological effect. You can see that Ki67 does drop with exposure to this drug to a greater extent than anastrozole alone. And this seems to be particularly true in the luminal B subtype of hormone receptor positive cancers. Another part of the cellular machinery that could be amenable to improvements is the inhibition of cyclin-dependent kinases that directly regulate the cell cycle. CDK4/6 inhibition relies on an intact retinoblastoma RB pathway, and in preclinical experiments, the impact of CDK4/6 inhibitor with hormonal therapy was greatest in the luminal A and B cell lines. On the left side here, the bars in blue represent the ER-positive cell lines. The bars in red represent the basal cell lines. And you can see that most of the activity was in hormone receptor positive. So based on this, clinical trials have been designed to look at CDK4/6 inhibition. The first of these to be reported was a phase 2 study that showed dramatic improvement in progression-free survival with a hazard rate of 0.37. This is one of the largest impacts we've seen, although again, we have to be cautious. This is a phase 2 study. There's a higher error rate in this study. But nevertheless, this has led to considerable excitement with these compounds. There are several such compounds in the clinic now: palbociclib, LEE-01, and LY-235219. All of these are being tested. This one is with palbociclib. And a large phase 3 study has just been completed, and we anxiously await the results of this. This may be the first drug to be approved in this class, but we have to wait and see what the phase 3 data shows. The other area that I mentioned is epigenetic alterations in the methylation status of promoter regions and in the chromatin that affects gene progression. This is one of the key mechanisms of drug resistance and appears to be particularly true in hormone receptor positive cancers. In fact, in the Cancer Genome Atlas, when one maps all the methylated CpG islands, these are the islands, the areas that lead to gene expression, the luminal B subtype seem to have the greatest degree of hypermethylation. And on this basis, then, a histone deacetylase inhibitor, entinostat, was tested in refractory, so post-AI progression patients were treated with exemestane with or without entinostat, a histone deacetylase inhibitor. And this actually showed an improvement in survival. Again, this is a phase 2 study, and a phase 3 study is now on the verge of being completed to see if this might be another strategy to improve the benefit of hormonal therapy in the metastatic setting. So I've showed to you how the molecular biology of breast cancer in hormone receptor positive disease, both at the genomic level as well as at the epigenetic level, can inform us as to what pathways might produce better results when combined. And I think this is where the field of breast cancer is going for advanced disease and even for early stage disease, is that we classify cancers according to their biological characteristics and then develop combinatorial therapies that complement each other in eradicating the tumor. We call this synthetic lethality. That is, two drugs that alone may not be as effective, but together are more than double the effect. So hormonally sensitive disease can be defined by estrogen and progesterone positivity, but there's many other ways to define this. As you heard earlier, we can look at prior therapies. We can also look at gene expression profiling and other genomic features. And the benefit of combinations, as I pointed out, may depend on the details of these tumors. We may find a subclass that we can just use dual hormonal therapy, like an AI and fulvestrant, or signal transduction inhibitors, CDK inhibitors, epigenetic therapy. Of these, of course, the only one that is approved outside of the HER2 positive group is everolimus and exemestane. And we hope that this list will grow over time. Thank you for your attention.
Thank you, Professor Debio. We regret that there is no enough time for discussion.